Hi, my name is Chris Kramer, and welcome to Fetal Echocardiography, your course through CQU. Um, this is week one, and I'll explain kind of what we're doing, but just a little bit about me. I am a program director in Milwaukee, Wisconsin at Aurora Healthcare. So we've had an echocardiography program specific to cardiology services here at Aurora since 2010. Um, and I am the director. I started this program in 2010. We have grown over since, and we have this great partnership with your professors Sharon Kay, and I'm working with Ann Quinton on this course. So we are both co faculty on this course, and welcome. Really look forward to going along this journey with you and seeing all the different aspects of fetal echocardiography. Hopefully you find this course very instructive, and we'll be getting into a lot of different views in fetal echocardiography. So let's start with our first lecture, and this is week one, fetal echocardiography, human heart embryology, part 1A. So again, welcome to fetal echocardiography. We're going to learn about how the heart develops and how the cardiovascular system develops. And I really broke in week one into two lectures, and you'll kind of experience this, but they're two long lectures, part 1A and part 1B. Feel free to take a pause through this, and I put in some web links and some a reading material for you to follow along too. So try to make this interactive and fun for you too. All right, so here's kind of our outline. So human heart embryology. We're going to go through the cardiac morphogenesis, the cardiogenic fields, primitive circulation, the development of the heart, and then importantly the looping of the heart. And then at the very end here, we're going to talk about pericardial development before we move on to part 1B. And I just would like to start with this quote from Sal Bellow in his book Ravelstein, and it says, "The heart is a mystery. If you've seen your own heart on a video screen, as millions have done by now, convulsing and opening rhythmically, you may have wondered why this persistent muscle is so faithful in its function from uterus to its last breath. This rhythmic gripping and relaxing blindly goes on. Why and how?" So Sal Bellow was a Canadian-American writer, and as we look at echocardiography and we're looking at the heart in the womb, and then as going into adult life and with all the other as we talk about cardiac abnormalities, hopefully this will be very interesting, and we're in a great field, and think about this quote as we go on throughout the semester. So here we go. We have the cardiovascular system in embryology is the first system or first organ system to become functional, first system to develop and develop in the embryo. And so the primordial heart and vascular system appear in the middle of the third week. And we can actually visualize that by kind of day 18. Day 18, we can visualize by M mode and look at some of those things by ultrasound. And a lot of the research that we'll be talking about too is done on chicks and mice and looking at heart tubes developing. We'll go over that in a little bit, but you can see that there's a crucial time period that when the heart and are really an important time period when the heart develops. And this happens, anywhere between the 3.5 to 6.5 weeks post conception. And so the heart beats at about, anywhere between 22 to 23 days. And it is rapidly developing because the embryo cannot or no longer satisfy the nutritional needs of the fetus and oxygen requirements by this diffusion alone. So a pump has to be in place to circulate that blood, and that's where the heart comes into play. So I put together this kind of timeline that looks at human heart development. This is a great timeline to look at when things happen, and they're happening. We always think about when we're talking about one part, let's say the atrial septum, as we're going to talk about, and the ventricular septum. Well, they're going on at about the same time. And as we talk about different systems, keep that in mind. But this is a nice timeline just showing you from week 4 to week 10. Week 4, heart tubes fuse and loop. Week 5, this is when the atria and ventricles divide. Week 6, the atria start to septate. Endocardial cushions start to form. Truncus starts to divide. Week 7, the foramen ovale is the only opening. So blood is being shunted across from the right side of the heart to the left side of the heart, and we'll talk about that. And then we have, the kind of the vascular system of the heart forming, the coronary sinus forms, and then we have this division into our pulmonary artery and aorta, and then week 10, kind of our semilunar valves form. So we'll talk about all these and we'll break this down into the different timelines. I further kind of broke this down into looking at these embryologic events and looking at specific days, and this is very helpful too. The vascular tubes is formed kind of day 20. As we look at the muscular, let's say the septum and cushions. I also listed some of the defects that we could have. So on day 24, that's a day that the cardiac or the heart tube starts to loop. And if it loops the wrong way, then we get dextrocardia. So these are some of the, some of the defects that we'll be talking about further on in the semester. And again, important days to know as we talk about embryology, and this will come up again. Now, if we look at cardiac morphogenesis, it's very interesting if you've studied and gone through anatomy and looked at anatomy of other, let's say, animals. So you can see that a fish heart has one circulatory system, one heart, one atria, one ventricle, what we call as a single loop circulatory system. And then as we get into kind of warm-blooded mammals, or having these two parallel circulations, and this is what we have here. We look at most mammals, crocodiles, and birds have, again, a separate system, so pulmonary circuit, and then a systemic circuit. So pulmonary circuit goes to the lungs, oxygen, picks up oxygen, and then goes to the left side of the heart. That left side of the heart pumps to the rest of the body to get nutrients, oxygen and nutrients to all the organs, and then back through our capillaries and our return system back to the right side of the heart. And so we have two parallel systems. That's what we're looking at. That's what we'll be talking about in embryology. Just another kind of a look here versus the single loop system versus the double loop system. Interesting enough, reptiles, you can see there's a mixing of blood. They do have a double loop system, but there's a mixing of blood. And here, not a closed system that we're mammals and what we're used to seeing here. But interesting to really understand that morphogenesis, and really, again, we're not going to get into fishes and reptiles, but it's very interesting just to look at how these pumps work and how these pumps develop. So really, when we talk about that morphogenesis, the heart develops, that there's predetermined kind of fates for all these animals, and they're all following the similar plan. So the heart develops following the same general pattern in all vertebrates from fish to humans, and it starts with this fusion of the myocardium and the endocardium to a single tube that starts to function. Then there's looping, and then followed by chamber specification. Now the heart chamber, or the heart, needs a conduction system to time out the different events, right? As we talk about cardiac events, which we'll talk about more in the semester too. There has to be coronary circulation, and then valves form too. So it follows that same pattern, whether it's a fish or a human, and then we go into those different loop systems. So as we said, the primordial heart and vascular system appears in the middle of the 3rd week. And as we look at this kind of schematic, this is a schematic taken from the developing human, and we'll be referencing a lot of these beautiful pictations or pictures from that. And as we look at this tube, here's our heart tube, right? We can see our umbilical vein, umbilical cord, umbilical artery. Okay, so getting blood, and we'll talk about the circulation in utero, right, or fetal circulation, getting blood from the mother, and how that goes in through the fetus, and the development of that. So we're going to start talking about this heart tube and how it's formed too. And so that brings us to our cardiogenic fields. So our cardiogenic fields are the earliest precursors of the heart and are located in this bilateral paired cardiogenic fields. They're located in the splanchnic layer of the anterior lateral plate mesoderm. And when these cardiogenic cells form, both the endocardium and the myocardium come together. Plus there's two non-cardiac populations, a dorsal mesocardium and a parietal pericardium, and that forms within these cardiogenic fields. Now there are certain regulatory genes and factors that we rely on for cardiac morphogenesis. If you're really interested, there's a great book called Cardiac Development. It's by Margaret Kirby, really going into all the different genes and regulatory or transcription factors going into heart development. But if we break it down, we have bring it back to that cardiogenic fields. We have these two bilateral troughs in the myocardium. They're cradling that endocardial tubes. These tubes are brought ventral or midline during the closure of the ventral foregut, and it kind of shapes this W, and we'll take a look at this here too. And then each tube is continuous with the first aortic arch as they come closer and closer together. So here's what that W kind of looks like. All right, so here's heart tube. They come together, right? So it's two tubes coming together. It's forming this foregut, right? And these two layers come together. If we look Closer here, again, this is a cross section of the fetus here, and we can see as it develops, these two heart tubes come together, forming one. As they form one, we'll talk about this too, but they form one heart tube and surrounded by this cardiac jelly, but with that forms the myocardium and endocardium. All right, so here's just another view as this forms. Right, here's our heart tube. Our heart tube forms as this anterior portion comes up and kind of hooks or comes then inferior, forming, this would be the head or forebrain, and you can see how the heart is kind of moved into a kind of very central location here. So on day 22, the heart tubes merge and fuse. This forms the primitive heart tube. Circulation begins and the heart starts to beat. So 22 to 23 days, depending on the literature that you read, is when the heart tube starts to beat. The tube becomes endocardium at this point, and that mesoderm becomes the myocardium and epicardium, and so really forming the three layers of the heart. So endocardium was the innermost layer, myocardium is kind of the middle section, right? And the epicardium is the outermost layer of the heart. And throughout this process, the myocardium secretes this thick acellular matrix called cardiac jelly. And this cardiac jelly forms a layer separating the myocardium and the endocardium. So cardiac jelly is an important part of embryology, or forming this heart tube. And here's that cardiac jelly that we were talking about. So here's the two tubes. Here is the cardiac jelly when it forms one tube. Again, these are coming together. These two, we'll show a picture here in a second, of two tubes coming together and forming one single heart tube. So with this, we just talked about kind of the cardiogenic areas moving towards the head of the embryo. And in this next phase of the heart development, this heart tube narrows and lengthens. And so we get into, again, this primitive heart tube. Heart tube appears, again, by the third week, and as you know, it begins to beat by day 22 to 23. And really, there is kind of this unidirectional blood flow that occurs in the primitive heart tube, and it comes from kind of inferior to superior, or from the foot to the head of the fetus. And we'll talk about how that changes, but there is this, again, in this tube, there is this unidirectional blood flow. Again, the heart completely is formed by the week 7, and is the first organ to fully develop in the fetus. And we have these kind of initial contractions of the heart, and they start within the muscle, or starting within the muscle, this myogenic origin, in and starting from the muscle. But blood is moving through that tube. And so the muscle layers of the atrium and ventricles outflow tract are continuous. They are continuous. Again, there's no valves that are forming. And we have these kind of wave-like contractions. And that is starting kind of in the sinus venosus, a part of this heart tube, which we'll go through. And at first, circulation through the primordial heart is this ebb and flow type. But then by the end of the 4th week, there's this coordinated contractions of the heart. And this results in this unidirectional flow. So at first, it's kind of, as it's pumping, it's going through kind of the wave-like, and then becoming this unidirectional flow. We're going from kind of the base, or from inferior to superior. So we do have veins that are associated with the embryo. During this time, the blood vessels are also forming as the heart is forming, and this forms within the wall of the yolk sac of the embryo. The vessels are eventually joined together to form the primitive cardiovascular system, and we have these three paired veins that drain into the primitive heart tube through the sinus venosus, and we have these common cardinal veins, umbilical veins, and vitelline veins. And so the vitelline veins return poor oxygenated blood from the yolk sac. Umbilical veins bring oxygenated blood from the placenta. And the common cardinal veins return poor oxygenated blood from the embryo body to the heart. And again, all of these form later in life into different aspects. And here's this yolk sac, right? So the vitelline artery is here, vitelline vein, and then comes back into the sinus venosus. Placenta would be here, so this is the umbilical vein, umbilical artery, right? Umbilical vein, again, coming to the sinus venosus. Again, here's the primordial heart, heart tube, and then goes to kind of these or arch arteries to the rest of, again, the fetus or the embryo. So veins associated with the embryo, as we continue on with that, blood actually starts to form during the fifth week. These vitelline veins enter the venosus end of the heart, or sinus venosus end of the heart, and there's a left and right, and it's important that we kind of know what they become. But the right vitelline veins become the hepatic veins and portal veins, into later the IVC, right? And then the left vitelline veins regress and disappear. The cardinal veins are really the main venous drainage system, and there's a anterior, a common, and a posterior cardinal vein. These form, kind of goes through the same process, but the right cardinal vein forms the sinus or superior vena cava, and then from the other cardinal veins form the IVC, azygous vein, hemizygous, iliac veins from those cardinal veins. Remember that our umbilical veins supply the oxygenated blood from the placenta, so from the mother. And we have this large shunt that develops between the liver and the umbilical vein called the ductus venosus. And we'll talk more about that when we talk about fetal circulation. So as we go through and we go through our two tubes forming a single tube, right? So we can see two separate tubes as that W, as we talked about, forms and these two tubes come together. They form a single tube, right? And we'll talk about all the different functions. So again, just showing, here's our left vitelline vein that regresses. We do see that, again, remember, blood flow is from kind of this inferior to superior portion, right? So it's unidirectional. It's going in this motion, right? So as we look at this motion of blood flow, just take a look at this top. We'll talk about the folding and the looping of the heart here next, but that will be the bottom part of this diagram. So again, just looking at the veins associated with the embryo, review those: vital line veins, umbilical veins. Here's that yolk sac here, placenta, right here. Here's those anterior common and posterior cardinal veins, again, that form, again, as we talk about SVC, IVC later on in development. That's what they're forming in the embryo. These veins are associated with the embryo. So again, let's discuss the branches of the dorsal aorta. And for now, as we talk about the fate of the aorta and the fate of the aortic arches and the branches, we're just going to talk about kind of the umbilical artery for now. So umbilical arteries carry the poorly oxygenated blood back to the placenta. Those become the iliac arteries, and those vital line arteries, again, they feed the primordial gut. And as we talk about branches off the aorta, they become the celiac artery, so the superior mesenteric artery, and the inferior mesenteric artery, or SMA and IMA. So later on in development. But as we go through this primitive heart tube, even though there's contributions of the left and right side of the heart, they're forming these regions are strictly to the left and right sides of the cardiac tubes, and both are contributed to both ventricles. So meaning that these two tubes, as they come together, as they form one single tube, they become parts of the left and right sides of the ventricles. And the left one doesn't say that, left one, it makes the left ventricle, they form, and as they form, they contribute to really both sides of the left and right side of the heart. So again, looking at these two tubes, looking at the structures, now we can see that as they form, again, they contribute to all parts of the heart, and going through both contributing to the right side and left side of the heart. Okay, so let's talk about the development of the heart. And really, there's 5 different regions that I want you to remember. And these kind of grooves develop within the primitive heart tube, and they divide into these 5 sections. And if we think about, this is anterior to posterior in this view, but if we think about it as superior to inferior, and that's, I like to always kind of point out that way, as we look at these different diagrams, you can see, again, 5 distinct sections or regions. And all those have names. So we have the sinus venosus with this section, the atrium here, ventricles here, or ventricle, the bulbus cordus, and then the truncus arteriosus. And as we talked about with, the heart tube forming and blood flow, again, it's going to be coming from inferior to superior, or in this picture, posterior to anterior, but it flows through the sinus venosus, then through the atrium, through the ventricle, to the bulbus cordus, and then through to the truncus arteriosus, and then to the aorta here, right? So as we're going through, or the aortic arteries or arch arteries. So it's very important to kind of know those. Again, all sections here, these 5 distinct layers, because we're going to talk about how they form and what their role is in part of their developing into other structures. So the sinus venosus, what does that become? The truncus arteriosus, what does that become? And so I'm going to give you, we're going to break this down into into sections. And again, this is another picture. This is from that Developing Human, chapter 13. And as we look at this, again, blood flow coming from really this inferior portion flowing through in this unidirectional forming or unidirectional motion, and through to the truncus arteriosus. And so sinus venosus and the atrium, right? Here's our primitive ventricle. Here's the bulbus cordus, and then the truncus arteriosus. And I like to throw this in here, but I'm not sure if the truncus arteriosus is an embryology term or a Harry Potter spell. Kind of sounds like a Harry Potter a spell here. So I'd like to throw in a little humor every once in a while. All right, so here we go. So truncus arteriosus. So if we start from the most superior portion here, this truncus arteriosus forms the aorta and then forms the pulmonary artery trunks. And just if this fails to form, if there's a failure of the truncus to divide, and we'll talk about that later, if we have failure to divide, then that can result into a defect called a persistent truncus arteriosus. Well, what about the bulbus cordus? So the bulbus cordus can be divided really into three sections. The bulbus cordus becomes the primitive right ventricle. It contributes to the ventricular outflow tracts, or the conus cordus, and portions of this kind of truncus arteriosus. Kind of again, if you think about the outflow tracts of the heart, the RVOT and the LVOT. RVOT being the right ventricular outflow tract, LVOT being the left ventricular outflow tract. And so if we have failure or defects that can form in the bulbus cordus, so we can have these conal truncal abnormalities, including truncus arteriosus, in something called or tetralogy of Fallot, or TET for short, tetralogy of Fallot. We'll talk about that more during the semester. Let's move to this ventricle section, or the primitive ventricle, what we like to call as the morphologic left ventricle. As the heart shifts towards the left during development, as we talk about looping, it becomes the left ventricle. Eventually, this will divide, and this ventricle will have an interventricular septum that forms between the right side of the heart and the left side of the heart, specifically the left and right ventricle. And if we have issues with the primitive ventricle, or this section of that heart tube, this is where we can have a univentricle heart, and that's about 80% of univentricle hearts consist predominantly with a defect in this morphologic left ventricle, which makes sense. Next section would be the atrium. So again, the atrium will divide. This primitive atrium will divide into will divide equally into the right side and left side, and it divides by an interatrial septum, and that septum forms a septum primum, a septum secundum, and then a foraminal valley, so that there's blood flow that can get from the right side to the left side of the heart. Again, forming that primitive atrium, forming the left atrium and the right atrium. And if we have defects there, again, that would form atrial septal defects, and there's a number of different atrial septal defects that we'll talk about throughout the semester. And then lastly, the sinus venosus. So the sinus venosus really consists of two horns of the sinus venosus, here and here, left and right. They contribute to the formation, as we were talking about, with the SVC and the IVC, or that return system of blood to the heart. And it's important to know that the coronary sinus, or the CS for short, forms on that posterior wall of the atrium. And the coronary sinus, again, is bringing blood back to the right side of the heart after it's been fed by the coronary arteries. So it's a return system to the heart, and that's formed in the sinus venosus. Now, if we look at the right and we talk about the right and left horns of the sinus venosus, the right horn forms the posterior wall of the right atrium, the eustachian valve, and the Thebesian valve. The Thebesian valve being the valve that is in the ostia of the coronary sinus. If we look at the left proximal part of the left horn creates that coronary sinus, and the distal part of that left horn kind of disappears. But if it persists, we can have what's called a left SVC, and that left SVC then enters the coronary sinus and or the left atrium. So blood flow we can have a problem with blood flow from that left SVC, either entering the coronary sinus, making that dilated, so we have an abnormal route for blood flow, or it can actually shunt blood from the return system into the left side of the heart, causing problems. Now, we do have one other part between the primitive atrium, and the primitive ventricle, and that's the atrioventricular canal. And that's a large communication between the primitive atria and the primitive ventricle, and that again forms these endocardial cushions. And as we have these endocardial cushions, they divide into two atrioventricular orifices, and those orifices would be, on the left side would be kind of the mitral valve, on the right side would be the tricuspid valve. And if they don't form correctly, we get these endocardial cushion defects. And as we talk throughout the semester too, this is very common in Down's syndrome patients. And so they have an atrioventricular canal with shunting of blood between kind of the atrium and the ventricles too. So here we go through the circulation through the heart tube again. As we talk about all sections of that heart tube, blood enters through the sinus venosus, then blood moves from the sinus venosus and enters into the primordial atrium, and flow is controlled by the sinoatrial valve. And I'll show you a picture of that here in a second. Then blood passes through the atrioventricular canal, or AV canal, into the primordial ventricle. And when that ventricle contracts, blood is pumped through the bulbus cordus and the truncus arteriosus into the aortic sac. Again, and from there, from the aortic sac, it's distributed through the pharyngeal arch arteries and the pharyngeal arches. Here is a picture of that. Again, here's the sinus venosus, primordial atrium, AV canal, primitive or primordial ventricle, bulbus cordus, and then truncus arteriosus, going through the aortic sac, and then that blood flow distributed through those pharyngeal arch arteries. So again, coming through, again, as we talked about some of those vascular parts that form, right? Here's that common cardinal vein into the sinus venosus. Here's that sinoatrial valve. Again, not in essence a valve that we think of seeing like the mitral valve or tricuspid valve or any semilunar valves. Semilunar valves would be, again, your aortic valve and pulmonary valve. Again, no no really valves forming, but these kind of canals forming here. AV canal. Okay. Remember that the primitive heart tube, as it forms, both sides of the heart tube contribute to both of the ventricles, or both sides of the heart. And heart differentiation does not depend on the heart, or that single tube, or that single tube convergence as those two hearts form, but the hearts, meaning the different segments of the heart, the formation of the left ventricle, left atrium, right atrium, and the AV canals form due to this looping process. And as we look at the bottom section from this, the developing human diagram, we start to see this looping of the heart from D here, this picture here compared to this picture, and we can see all the segments that we were talking about: sinus venosus, the primordial atrium, primitive ventricle, the bulbous cordus, and the truncus arteriosus. And as we loop, or as the heart loops, we can see that it takes on a different structure, and we're going to talk about that through the next slides here. And as we talk about the development of the heart, remember that there's an atrial end and a venous end. And those two tubes, or those ends of the tubes, are surrounded by this layer of visceral pericardium. And so as we talk about the formation of these tubes and the chambers, remember that the pericardium is surrounding those tubes. And so here is, if you look, here would be the pericardium in this picture here. Here's the pericardium, so within the pericardium, or visceral pericardium. So the primitive heart tube then elongates within the pericardial cavity, and the bulbous cordus and the ventricle grow more rapidly. And with that, the growth of those two, the bulbous cordus and the ventricle, those two form, and as they're growing, form this kind of, the heart takes on this kind of U shape, or later an S shape. And because they're within a tube, within that pericardium, there is this twisting or looping that forms. And so here's a picture here of the pericardium, and if you think about it, attached kind of at the superior and inferior portions here within this pericardium, we can see that the bulbous cordus and the ventricle grow, and as they grow, they kind of form this U shape. And here's that kind of U shape, and we'll talk about later this S shape. And just notice what's happening to this bulbous cordus going inferiorly. Look what's happening to the sinus venosus. That's moving more superiorly. Okay. So as the heart tube begins to loop, at 4 weeks gestation, the tube rotates 180 degrees, and the heart tube loops anteriorly and rightward, creating this bulboventricular loop. The opening between the primitive ventricle, or the left ventricle, and the bulbous cordus is called the bulboventricular foramen. And as we look at that looping, the looping creates this broad outer curvature and a narrow inner curvature. So we'll look at those two, and these cavities of these chambers are formed. When they're formed, there is a growth of that outer curve versus the inner curve. And there's a TGF beta factor that takes our nodal factor that is involved in the looping of the heart. So there is some genes and genes and growth factors that are involved in the looping of that heart tube. Here again, if we see the ventral view, here's a ventral view and side view, but we see this outer curve and inner curve, right? So now we have, again, if we're looking at this, again, looping to the left side, right? Or moving to the left side as this sinus venosus starts to move more anteriorly. But again, there's a rotation of this tube to the right. So if we look at it from this ventral or frontal view, we see that go to this tube go to the right side of the patient, right? Patient's right. And as we see that tube, we start to see this bend, so the outer curve versus the inner curve, and again, then sinus venosus becoming more, again, anterior or superior, superior in this diagram. And if we go back to our segments of our heart tube, you can see that, again, the truncus arteriosus pretty much stays in that same spot. And as it shifts towards the right, so from that single heart tube, elongated tube, we shift that to the right, so the ventricle, bulbus cordus, and ventricle start to move down, and as the atrium and the sinus venosus start to move more superiorly. And we do see this in chicks, so this is looking under a high power microscope at chick development. And as we talked about before, as we talked about that cardiac morphogenesis, we can see that forms this, our heart tubes form. Here's these two tubes coming together, these two tubes forming a single tube. The single tube, again, sinus venosus to truncus arteriosus. And then as that outer curve towards the right and the sinus venosus starts to move up more superiorly, we get this formation or looping of that heart tube. So again, this would be the frontal plane or ventral plane. This would be kind of that side view, what's happening, and we'll go step by step through this. So the primitive atrium and the sinus venosus, as we talk about looping, move superiorly and posteriorly. Okay, so superior and posterior, right? So it's going to move that sinus venosus and atrium, or sinus venosus and primitive atrium are moving superiorly, but they're also moving posteriorly. Okay, so this would be anterior plane, posterior plane. That primitive ventricle then moves to the embryo's left side. So here's this primitive ventricle, right? So bulbus cordus, remember, is moving down. Ventricle, bulbus cordus and ventricle here, but bulbus cordus moving towards the patient's right. So that bulbus cordus, again, bulbus cordus moves inferiorly, so coming down. Anteriorly, patient, here's anterior plane, and then again, as we said before, just towards the embryo's right. Okay, so know again all sections of that heart tube and how they form. And so the primitive atrium, just in kind of wrapping this up, the primitive atrium and the sinus venosus move superior and posteriorly. The primitive ventricle then moves to the embryo's left side as the bulbus cordus moves inferiorly, so comes down, and anteriorly to the embryo's right. And there's a great resource here. I want to recommend that you go to this Indiana University website. You do have to have Firefox to run this, and they recommend running this on Firefox. Firefox is a free downloadable web browser that you should be able to get off the internet. And I'll put a link to Firefox as well. And through these websites, it helps you kind of walk through again the primitive or the looping of the valves or looping of the chambers. So I would encourage you to go to this website, and we'll take a look at it. I'll bring it up in the next slide here. And so this is what the website looks like. This is that Indiana University website. And so as you look at this, there's things that we will use throughout this next two kind of lectures and into week 2. But here is, again, you can go through the folding, the early heart and primitive heart tube folding, overview of the folding and the early heartbeat. And so I recommend going through there. Really, it's very nice. They do give a really nice overview. They'll give you some questions here too, to kind of do a pre-test, post-test, see where you're thinking as you're going through this, and hopefully you get these right. And we're not going to score you on this. This is just to test your retention as you go through the animation. So I recommend that you go through these at your leisure. This is a great website, and as we look at it too, it will be on your Moodle site, and it will be linked there. Well, as we talk about heart looping, what are some defects that can happen as early as the heart looping? Well, if we look at dextrocardia and how that happens, look at this diagram. So normal looping, we'd have the truncus, here's all the segments here, and instead of going to the bulbus cordus and the ventricle, here, bulbus cordus, instead of going to the patient's right, what if it goes to the patient's left? And if the heart tube bends itself left instead of right, we get this, what's called dextrocardia. So now the apex of the ventricle is going to be opposite. And so the heart will be displaced to the right, the vessels will be reversed, it'll be kind of a mirror image. And dextrocardia is the most frequent positional defect of the heart. So it's everything in reverse, and that happens again if there's instead of bending that heart tube to the right, it bends to the left. So the last section here is really talking about the pericardium, and we have to go all the way back to the cardiogenic fields. So let's take a step back and look at that primitive streak, and we talked about that primitive streak, and we talked about where those cells form. Really what we want to talk about is the pericardium. And so as the second part of that second bullet, there's two non-cardiac populations, the dorsal mesocardium and the parietal pericardium. The dorsal mesocardium disappears, and this parietal pericardium forms. And this parietal pericardium, or this pericardium, surrounds the heart. And so at the 5th week, there's this pleural pericardial fold, and it begins to migrate towards the midline. And as the migration forms, it's pulling along these phrenic nerves. So these phrenic nerves travel with this pleural pericardial fold as they migrate down or migrate midline. And then what happens during pericardial development is the root of each fold travels ventrally as the pleural pericardial folds, and that grows medially. And so we can see, again, here's the heart tube. As those two pleural pericardial folds come together, we can see that they surround the heart. So this would be surrounding the heart, the pericardium surrounding the heart. And again, there's as we look at the different structures or different that website from the University of Indiana, I encourage you to look at the pericardium and how that develops too. There's another nice picture with a pre-test, post-test. So at the end of the 5th week, that those pleural pericardial fuses or folds fuse, creating a thoracic cavity and a pericardial cavity, and there's and two partially formed pleural cavities. And so here would be again surrounding the heart, surrounding that tube, heart tube. Here would be the pericardium. All right. Here's the pleural cavities. You can see the lungs developing here. Okay, so I would encourage you to go to that website too. And this is, again, just how the pericardium forms. Here's a list of resources that I use throughout this presentation. A lot of information coming again from that Developing Human resource, chapter 13, which is the circulatory system or formation of the heart. Echocardiography in congenital heart disease made simple, and then echocardiography in pediatric in congenital heart disease from fetus to adult. Now, recommended reading, of course, from your book that you purchased for the course in the Practical Guide for Fetal Echocardiography, and that's chapter 3 for this lecture in week 1, going through really embryology and formation of the heart, and then other 2 recommended resource readings. So that is it for part 1A